Good morning, children.
Morning, ma'am.
We have completed the chapter Plan, Growth and Development. Today we are going to start with a new topic. And a topic for today is about a process which provides food for all the living organisms on the earth. So if this process will not take place, none of the organisms on the earth will get food. Can anyone tell me about which process I'm speaking about? The process?
Photosynthesis.
Yes, photosynthesis. And photosynthesis is carried out by plants and it is a source of food for all the living organisms. Either directly or indirectly, we all are dependent on plants for our food. And how plants prepare food, or one of you define what is meant by photosynthesis. Steffi, define photosynthesis.
The process by which plants absorb the sunlight, carbon dioxide, and water to make food.
Okay. That's it. So you can define it as the process by which green plants prepare food using carbon dioxide and water in presence of sunlight. So that is the definition you have learned from a very small class. From which class you have started learning photosynthesis? Class 6 or even before that? I know you all are familiar with the process. So the topic of this lesson is about photosynthesis. Okay, let's continue. Photosynthesis. Photosynthesis is a physico-chemical process. Here you can find, or here light energy is getting converted into chemical energy. Solar energy or light energy is getting converted to chemical energy. So it's a physico-chemical process by which green plants use light energy, that is solar energy. Solar energy is a source of energy for all living organisms. So it's the ultimate source of energy. So utilizing light energy, plants synthesize organic compounds. Raw materials like carbon dioxide and water are also essential. So it's a physico-chemical process by which green plants use light energy to synthesize organic compounds. Organic compounds, carbon containing compounds like carbohydrates. Photosynthesis is the basis of life on earth, because if photosynthesis is not there, food production will not take place. Without food, none of the organisms can survive. So it is a basis of life on earth. And apart from preparing food, photosynthesis is having one more significance, or it is significant in yet another aspect. Can anybody tell me what is the other importance, one main significance of photosynthesis? Definitely it is helping plants to prepare food and provide to all the other living organisms. Apart from that, something which is essential for our survival is also being produced during photosynthesis. What is that?
Oxygen.
Oxygen. So without oxygen, we cannot survive. So oxygen, the life-saving gas, your breathing gas, plus food is provided by green plants. Ultimately, all living forms depend upon solar energy, sunlight for energy, because we all are dependent on plants for our food, and plants are dependent on solar energy for preparing food. So this energy only, light energy only, they are converting it into chemical energy. So directly or indirectly, we all depend upon sunlight for energy. Now, what is the significance or importance of photosynthesis? Primary source of food for all living organisms. It also releases oxygen into the atmosphere. It purifies the air. Once we are breathing, once living organisms breathe out, we'll be releasing carbon dioxide. We take in oxygen for breathing and give out carbon dioxide. This carbon dioxide, if it fills the atmosphere, that can pollute the air. So plants absorb these carbon dioxide molecules and convert it into oxygen molecules by the process of photosynthesis. Now there are different experiments. So since very olden times, several experiments have been carried out related to photosynthesis. So we'll be learning about some of these experiments and some of the scientists who carried out different experiments related to photosynthesis. So let us start with variegated leaf experiment. This you had learned in class 10 as an activity. It was given in the chapter life processes. And variegated leaf means the leaf will not be green throughout. It won't be uniformly green. Instead, on the leaf, you can find green patches along with other colors. It can be yellowish, purple, different other colors can also be seen. And only certain areas will be green in color. As you can see in the figure, the variegated leaf given here has got yellow and green patches. Take two leaves. Two leaves should be taken. A variegated leaf or a leaf which is partially covered with black paper, and either if a variegated leaf is not available, you can take a leaf, cover it partially with a black paper to prevent sunlight from falling on it, and you can take it. And the one that was exposed to light. So the leaf which was exposed to light can be taken, and then what we do is test these leaves for starch. How to perform the starch test? Nandana, tell me how to perform the starch test. What is the method? Nandana is there? Nandana,
Yes, ma'am.
how to perform starch test?
I don't remember.
Final step.
I
Huh?
don't remember.
You should remember. You are learning it from class 7. You try. I will help you. Make it try. Rest, it is wrong also, no problem. Nandana, start. I'll help you. First, what we need to do, we'll pluck out the leaf, and then what is the next step? Where did she go?
Pure only, ma'am.
Then tell me. Why your videos are off? In the beginning itself, I told everyone's video should be on. Michael, Anthony, Naureen, Malavika. First, what we need to do, Nandana, we have to boil it in water. Then, after boiling in water, yes, first we need to boil it in water. Is there anyone who can help her? Nobody. Starch test, children, class 7 textbook it is given. Last year, in 10th standard also you have learned starch test. Nandana, I didn't expect this from you. Try, no? I told you to try. First you have to boil the leaf in water. Then after taking out from the leaf, you need to boil it in what?
Alcohol.
Alcohol. Then Nandana, unmute. Then it should be boiled in alcohol. Why do you boil it in alcohol? Why we are boiling it in alcohol? I have to check whether you people I'm teaching 11th standard or not. This I didn't expect from you children. Gayatri, after boil, why we are boiling it in alcohol? What is the intention? Or upon completing boiling in alcohol, what change you notice in the leaf?
The chlorophyll,
Correct. Chlorophyll will
they'll fade, something like that. And we'll put iodine.
louder.
Then we'll put iodine. And
When you are boiling it in alcohol, what will happen to the leaf? Leaf will remain as such. Will there be any change in the color of the leaf?
the chlorophyll parts will be
Saying is correct. Complete it. The chlorophyll will get
they'll fade.
fading. See, when you reduce your voice, when you're not sure about the answer, let it be wrong. I told you you're free to make mistakes. The chlorophyll will dissolve in this alcohol. If chlorophyll is dissolving in alcohol, what will happen to the leaf? Leaf will still remain green in color.
No, the green color will be removed.
Color goes and it becomes pale. The leaf will become pale and the chlorophyll will become green the alcohol will become green in color because it gets dissolved in alcohol. Now what is next? We'll take out the leaf from alcohol. Then you will keep it in water for some time, because when you take out the leaf from alcohol, the leaf will be brittle. Brittle means once you touch, it can easily break. So to make it soft again, it will be kept in water for some time. So it will become soft. Now the last step. Nandana, can you tell me the last step for starch test? I'm not going to spare you. Which is the last step? The leaf is pale in color. Now what is to be done?
Iodine solution.
It has to be kept on a white surface and iodine should be added. If starch is, what color change you can notice? Michael, if starch is present, what will be the color change?
Purple, blue,
Louder.
like a bluish color.
Bluish black color. And if starch is absent, it will remain colorless. So that is starch test. So these leaves, we have to carry out starch test. So upon performing the starch test, you will find that only those parts which were green in color, will be performing positively for the starch test, or only those regions will change its color to bluish black, and those areas which were not green in color, they will not show any bluish black color. That indicates that chlorophyll is essential for photosynthesis. On a variegated leaf, yellow and green colored portions are there. So green colored portions upon performing the starch test will turn bluish black, indicating starch is present. Whereas the other part which is not green in color, that will not show bluish black in color. That indicates starch is not formed because no chlorophyll. This experiment proves that chlorophyll is essential for photosynthesis, or photosynthesis can take place only in the green parts of the plant. Okay? Is that clear?
Yes, ma'am.
Now moving on to the next experiment, half leaf experiment. Just a minute. So next one is about half-leaf experiment. So half-leaf experiment means, as you can see in the picture, half of the leaf or a part of the leaf will be enclosed in a test tube. Test tube or a beaker or in some device where KOH is potassium hydroxide, and potassium hydroxide soaked cotton. A cotton is dipped in potassium hydroxide and that will be placed inside a beaker or a vessel, and then half portion of the leaf is inserted into that beaker or test tube, or you can use any apparatus. And here you can see a bottle or even a test tube can be used. So half of the leaf will remain outside, but half of the leaf will be kept inside a test tube or a vessel containing KOH soaked cotton which is dipped in KOH, that is placed inside the vessel, and half portion of the leaf is inserted into the beaker. Then what happens? What about the conditions existing outside and inside the beaker, children? So sunlight, water, all these are provided. Even water will reach that other part of the leaf which is inside, because the roots are transferring it to all parts of the plant. It is sunlight is also present, but something is lacking in the portion of the leaf which is exposed to KOH. Can you tell me what is lacking? This you have learned in class 10. What is special about KOH? Can anyone tell me what is special about KOH? KOH is capable of absorbing what?
Carbon dioxide.
Carbon dioxide. So the portion of the leaf which is inside the beaker will not get carbon dioxide, because the carbon dioxide which is present in the air inside that vessel will be absorbed by KOH. So carbon dioxide will be lacking. All the other conditions are common for the portion of the leaf both inside and outside. Then now you will test the leaf for starch. So then you will find that the portion of the leaf which was outside, which was exposed, they will show the presence of starch. Whereas the portion which was inside, it shows negative starch test, no starch formation, which indicates that no photosynthesis took place. Why no photosynthesis took place? Because carbon dioxide is essential for photosynthesis to take place. So carbon dioxide was lacking. This proves that carbon dioxide is essential for photosynthesis. So I'll repeat it once again. The first experiment was to prove that chlorophyll is essential for photosynthesis. Second experiment is to prove that carbon dioxide is necessary for photosynthesis. And the experiment, you call it as half-leaf experiment. Why it is called so? Because we select a leaf from a potted plant. Part of the leaf will be enclosed in a test tube containing KOH. This KOH can be soaked in a cotton and placed inside the vessel. And the KOH will take up all the carbon dioxide which is present in the surrounding air, which is present inside the vessel or in the apparatus. And after keeping this leaf in sunlight for some time, if you perform the starch test, you will find that the portion of the leaf which was exposed, which was outside the vessel or outside the apparatus, shows positive starch test, or starch was formed, photosynthesis took place, starch formation took place. Hence, they will show positive starch test, or they will turn bluish black in color. But the region which was inside the apparatus, it was not having carbon dioxide. It was not getting sufficient amount of carbon dioxide since it got absorbed by KOH. So that portion will remain colorless upon performing the starch test, which indicates that carbon dioxide is essential for photosynthesis. Now we are moving on to see another set of experiments, some early experiments performed by some eminent scientists. So we will start with the experiments carried out by Priestley. Have you heard about Priestley before in chemistry? Anybody? Children, have you heard about Priestley before?
No, ma'am.
Children, I told you something in the beginning. I had asked you to turn the videos on. See, children, if you're not interested to do that, you just attend the recorded session. That is same as this one, no? Hiding and attending the class. It is, you are not interested in interacting. That means you just attend the recorded session. Don't waste your time. You can watch the recorded session at your convenience. So choose that. But I insist, for me it is very particular that you have to turn the videos on unless you have some genuine problems. Otherwise you may not attend. You are free not to attend. Michael, what is your problem? Your connectivity was very good in the beginning. Suddenly what happened? And Nia, who gave me a message? Okay, two of them, Malavika and Nevin, informed me. But what about others? Now don't come up with the excuse that we all are having connectivity issues. And I told you why I'm insisting you, there is a reason behind that. And still continuing the same thing. If smaller class, if children who are not knowing about all that, I can understand. You are grown up children. You should not behave like this. If you don't want to be present, then leave. Now also you can leave. It's at your, just a click on the leave button. You leave the class and go. Recorded sessions you can watch, no problem. Where have you heard about Priestley? Can anyone tell me where have you heard about Priestley? He discovered something. Very, very important molecule. Nobody knows. Can anybody tell me? Nobody. He discovered oxygen in 1774. You might have learned it in chemistry. He performed several experiments to prove the role of air in the growth of green plants. So his experiments were mainly focused on proving that air is very essential for the growth of green plants. And he is the person who discovered oxygen in 1774. And he performed a very classic experiment where he tried out burning a candle in a closed place. Here you can see some bell jars. So bell jar, because of its peculiar shape, you call it as a bell jar. Inside a bell jar, he kept a lighted candle. And what he found was after some time, the fire got extinguished. The candle stopped burning after some time. It was burning for a while, then it stopped suddenly. And he tried out then he tried out to keep a mouse inside. A mouse was kept inside along with the candle. Still, after some time, he found that the fire got extinguished and the mouse died. Then he found out that, or he concluded that, a burning candle or the breathing animal is damaging the air. So some damage is happening to the air when animals breathe and when a candle burns. So that is what he understood. So in the beginning, he just lighted a candle inside and kept inside a bell jar. After some time, it got extinguished. Then he introduced a mouse into it along with the burning candle, and this candle got extinguished and the mouse died after some time. And he felt that, or he concluded that, a burning candle or a breathing animal damaged the air. He placed a mint plant. He placed a mint plant inside the same bell jar. Along with the mouse and the candle, lighted candle, he kept a mint plant inside. And to his surprise, he found that the mouse stayed alive, candle also continued to burn. So in the first two cases, after some time, the candle got extinguished. But after keeping a mint plant, mouse didn't die. It continued to be, or it continued to live, or it stayed alive, and the candle also continue to burn. What could be the reason? He hypothesized that plants are restoring the air which animals and burning candles remove. So the air which was removed, the component of air which was removed by breathing animals and burning candles were restored by the plants. So what happened is, you know that when the candle is burning, for a candle to burn, oxygen is needed. If anything has to burn, it needs oxygen. So whatever air, oxygen was present inside the bell jar. Bell jar, it is a closed jar. There is no possibility of air from outside entering the jar. So what happened was, in the first two cases, the oxygen present was utilized by the mouse and the burning candle. And they gave out carbon dioxide. But this carbon dioxide was not exchanged, or this was not converted into oxygen, or there was no oxygen to replace this carbon dioxide. Or entire oxygen got used up and the bell jar got filled up with carbon dioxide. But when a mint plant was introduced into that, the candle, the burning candle, as well as the mouse they took oxygen and released carbon dioxide. But there was no chance of this carbon dioxide to get accumulated, because the carbon dioxide released by them, by the candle and the mouse, was taken by the mint plant. And the mint plant converted it into oxygen by means of photosynthesis. So a balance was kept between oxygen and carbon dioxide. That is why the mouse stayed alive and also the candle continued to burn. There was no shortage of oxygen because the mint plant was compensating for the oxygen which they took up. So as and when they are taking oxygen and releasing carbon dioxide, these carbon dioxide molecules were used as the raw materials by the mint plant to generate more and more oxygen by the process of photosynthesis. Any doubt? Any doubt?
No, ma'am.
No, ma'am.
Now the next, so we completed the experiments carried out by Priestley. Now moving on to the next scientist, Jan Ingenhousz. So his time period was from 1730 to 1799. He conducted the same experiment. The same experiments were carried out by whatever Priestley conducted. The same experiments were carried out by our Jan Ingenhousz, but the only difference what he made was he tried out the same experiment both in dark and light. In darkness as well as in the presence of light, he carried out these experiments. He showed that sunlight is essential. So as he demonstrated, as he tried out these experiments, both in darkness and in presence of sunlight, he could prove that sunlight is essential for the process to take place. So when this was carried out in the dark, he could not get the results. But when it was carried out in light, so the same results he too could attain. So he understood that sunlight is necessary for the plant for purifying the air, which is fouled by burning candles and also breathing animals. So he tried out both these, the same experiment in both conditions. He tried out it in light and he tried it out in darkness. So when he tried it out in darkness, what is the differential and why he is saying that plants need sunlight for purifying the air? Because without sunlight, photosynthesis cannot take place. Without photosynthesis, oxygen cannot be released. Without releasing oxygen, air cannot be purified. So when he tried it out in both conditions, in both situations, he understood or he concluded that for purifying the air, means for releasing the air, releasing oxygen, or for photosynthesis to carry out, sunlight is very, very essential. He conducted the same experiment. The same experiment was conducted using aquatic plant. So not only using a terrestrial plant, he also excuse me, he also tried it out with the help of an aquatic plant. So in this experiment, he found that some small bubbles were formed around the green parts of the plants. So when he performed this experiment in the aquatic plant, because it is an aerial plant, means a terrestrial plant, you cannot make out the oxygen being released. So it is assumed that oxygen is released, oxygen is being produced, but no visible signs of oxygen release you can find. But when you do it in aquatic plant, when the bubbles are released, these bubbles or these oxygen molecules will be seen as bubbles in water. Hope you understood the difference. When it is carried out on a terrestrial plant, oxygen will be released, but we cannot make out oxygen being released. But when the same thing is carried out in water, underwater, using an aquatic plant, the release of bubbles indicate that oxygen is being released. And in bright light, he could notice some small bubbles formed around the green parts. Green parts, while in the darkness, it did not happen. Because when he performed the experiment in bright light, he could see bubbles coming up. But when he performed the same experiment in darkness, he could not see any bubbles emerging. That is because these bubbles are actually oxygen molecules and it is produced as a result of photosynthesis. So in bright light, photosynthesis took place, oxygen was evolved and that was seen as bubbles. But in darkness, no sunlight, so no photosynthesis, no oxygen release and no bubbles formed. Later, he identified these bubbles to be of oxygen. Thus, he showed that only the green part of the plants release oxygen. And the bubbles he found only around the green parts of the plant. So, that also explains that photosynthesis can be carried out only by the green parts. Only those parts will release oxygen. So can we quickly summarize the points? Yes? Explain the half-life experiment. Steffi, please explain the half-life experiment. Not half life, half leaf.
A part of a leaf is put in a test tube containing KOH, and half of it is exposed
Why KOH is needed?
because it has the ability to absorb carbon dioxide. And then it's placed in the sunlight. Then after taking out, we noticed that the
Need to perform some test.
I don't know, ma'am. I have to check.
Is the test we have to do to find out whether photosynthesis is taking place.
Yes.
So upon performing the starch test, what difference you notice, Steffi?
The part which was enclosed, um, the part which was exposed will show positive for starch, and which was enclosed will show negative for
See,
starch.
they'll turn, they will not make any color change, they'll not show any color change, which means that no starch is formed. Okay. Then we learned about the experiments conducted by Priestley. So briefly explain the experiment carried out by Priestley. Meenakshi.
Kept a burning candle inside a closed bell jar.
Meenakshi, can you speak a little louder?
Priestley kept a burning candle inside a closed bell jar. And after some time, he found that the candle stopped burning. Then he kept a mouse inside the bell jar, and after some time, the mouse also was dead.
Okay.
And then he found that there is something which makes the atmosphere
Which is or which is changing the or polluting or removing something from the air. Then
and then he kept a mint plant inside the jar.
continue. Change he noticed. Inside the bell jar, he placed or he introduced a mint plant along with the mouse and a burning candle. What change he noticed now? Anna, unmute. Will it survive? Will it
Ma'am,
survive? Yes, so
he found mouse stayed alive and the candle also continued to burn
why?
because of the
What was the role
ox.
of mint plant?
Sorry, ma'am.
What was the role of the mint plant in saving the life of mouse and making the candle burn?
Oxygen is supplied. Oxygen is supplied.
How oxygen can be supplied by mint plant? It's having oxygen cylinder. How do the mint plant release oxygen? Very simple. Think about the topic, heading of the lesson. Carries out photosynthesis. So the products, the byproduct of photosynthesis, main product is carbohydrate, byproduct is oxygen. So once this oxygen is released, so there'll be no shortage of oxygen in the inside the bell jar. And the carbon dioxide released by the mouse as well as the burning candle will be taken up, will be cleared by the plants. Why plants need it, Anna? Why carbon dioxide is taken up by plants? What for?
Preparing their food photosynthesis, they need
Material for photosynthesis. Okay, we'll wind up. Thank you, children.